Hello everyone, welcome to the YouTube channel of Chameli Devi Institute of Pharmacy, Indore. In this presentation, we will going to discuss about one of the most important chromatographic technique, which is thin layer chromatography. Now, what is the objective of this session? Which points we will going to cover? First is the introduction part. Second one is the principle of the thin layer chromatography and its significance, solvent choice during the process, procedure how to perform the TLC, then retention factor value, which is also called as RF value. After that, troubleshooting TLC, which problems will occur during the process. Now, what is TLC? It is a chromatographic technique like other chromatographic technique. This is a method for analyzing mixture by separating the compounds in the mixture. It is used to help determine the number of components in a mixture, the identity of compounds and the purity of compounds by observing their appearance of a product or disappearance of the reactant. It can also be used to monitor the progress of a reaction. It is very sensitive technique which is used for the microgram quantities and can be analyzed in a very little time like 5 to 10 minutes. This is very simple, quick, time saving and inexpensive technique for the separation of compounds in the mixture. This technique is having two type of phases. One is mobile phase and other is stationary phase. Mobile phase is a suitable liquid solvent or a mixture of the solvent which is present in the chamber, and stationary phase is alumina or silica gel for the thin layer chromatography. Now, what is the principle behind thin layer chromatographic technique? So, the principle is depend on the separation technique. The separation relies on the relative affinity of compounds towards both the phases like mobile phase and stationary phase. The compounds in the mobile phase move over the surface of the stationary phase. The movement occurs in such a way that the compounds which have higher affinity to the stationary phase move slowly while the other compounds travel fast. Therefore, the separation of the mixture is attained. On the completion of the separation process, the individual components from the mixture appears as a spot at respective levels of the plate. Their characters and natures are identified by suitable detection techniques. Now, what are the significance of thin layer chromatography? Then, number of compounds can be determined in the mixture. So, if you run TLC using mixture of compounds, you can separate them as well as detect the number of compounds present in the mixture. You can use the number of spots developing on the TLC plate to approximate the number of compounds in the mixture. Second one is verify the identity and purity of a compound. Then this technique is also useful for the identity of the compound as well as identify its purity. Other than that, monitoring the progress of the reaction. Then you can monitor the progress and rate of reaction by performing TLC of the reaction samples at particular intervals. For example, while performing biotransformation of compounds, you can collect reaction mixtures at different time intervals and use TLC to monitor the conversion of the reactants to the product. Other than that, monitoring column chromatographic separations. So, this significance is when you can test the efficiency of column chromatographic separations by obtaining samples of the purified compounds and performing TLC. As an example, if you are trying to purify a pigment from an extract using column chromatography, you can analyze different illusions for purity by using thin layer chromatographic technique. Now, which kind of solvent should be chosen for the thin layer chromatographic techniques? So, for the beginners do not know where to start, what solvent should they pull off the shelf to use elute a TLC plate? Because of the toxicity cost and flammability concern, the common solvents are used as an hexane, petroleum ethers, and ethyl acetate or some esters. So here some examples are given for the expected illusion order of origin classes. So this is increasing polarity solvents which are move more slowly, they are mentioned here. Other than that, eluting power of the organic solvents like increasing polarity, they are more powerful eluters like alkane, toluene, halogenated hydrocarbons like methylene, chloride and so on. Now, the procedure of TLC, how to perform TLC or thin layer chromatography. This is having steps. In the first step, preparation of developing container. So, the developing container of a TLC can be a specially designed chamber, a jar with a lid or a beaker with a watch glass on the top. Pour solvent into the chamber to depth of just less than 0.5 cm to aid in the saturation of the TLC chamber with solvent vapor. You can line part of the inside of the beaker with filter paper, cover the beaker with watch glass and swirl it gently and allow it to stand while you can prepare your thin layer chromatographic plate. So, what is the significance of paper placement in the chamber? So, paper placement or air impregnation, it is an air impregnation process. You place your filter paper in the seal development chamber after the solvent but before the stationary phase. Remember that. It absorbs the liquid in the solvent and provides more surface area for evaporation. More surface area means more and faster evaporation. More evaporation means more solvent vapor in the chamber air, which is desirable. Second step is preparation of TLC plate. How to prepare TLC plate and how to draw a line on the TLC plate, what size of the TLC plate. So, in the TLC plate used in the organic chemistry technique are purchased as a 5 cm into 20 cm sheets. Each large sheet is cut horizontally into plates which are 5 cm tall by various widths. The more samples you can place to run on a plate, the wider it needs to be. Shown in the photo to the left is a box of TLC plates, a large uncut TLC sheet and a small TLC plate which has been cut to the convenient size. Handle the plates carefully so that you do not disturb the coating of the absorbent or get them dirty. Now measure 0.5 cm from the bottom of the plate using a pencil. Draw a line across the plate at the 0.5 cm mark. This is the origin, the line on the which you will spot the plate. Take care not to press so hard with the pencil that you disturb the absorbent. Under the line, mark lightly the name of the sample you will spot on the plate or mark number for time points. Leave enough space between the samples so that you do not run together. Above 4 samples on a 5 cm width plate is advised. Now, third step is spotting on the TLC plate. Then, first of all, you should know if the sample is not in the solution form, then you have to dissolve in a volatile solvent such as hexane ethyl acetate or methyl chloride. Then obtain a microcapillary. So dip the microcap into the solution and then gently touch the end of it into the proper location on the TLC plate. Don't allow the spot to become too large. If necessary, you can touch it to the plate, lift it off and blow on the spot. If you repeat this step, the weight area on the plate will stay small. For example, we have mentioned a picture of a spotted TLC plate. This example plate has been spotted with 3 different quantity of the same solution and is ready to develop. If you are unsure of how much sample to spot, you can always spot multiple quantities and see which look best. Now the fourth step is develop the plate. So we have mentioned here 2 pictures, initial process of the plate and when it should be removed. So place the plate, prepared plate in the developing beaker, cover the beaker with the watch glass and leave it undisturbed on your bench top. The solvent will rise up the TLC plate by capillary action. Make sure the solvent does not cover the spot. So you can see here in the second picture, solvent is run out below the half of centimeter throughout the plate. So allow the plate to develop until the solvent is half a centimeter below the top of the plate. Remove the plate from the beaker and immediately mark the solvent front with a pencil. After that, allow the plate to dry. Fifth step of the TLC is visualize the spot. So how to visualize? You can see here first image. In this image, if uh, there are any colored spots, circle them lightly with a pencil. Most samples are not colored and need to be visualized with a UV lamp. Hold a UV lamp over the plate. I have mentioned here a picture of UV light developed on the developed TLC plate. So hold a UV lamp over the plate and circle any spots you can see. But be aware, UV light is damaging both your eyes and your skin. So make sure you are wearing your goggle and do not look directly into the lamp. Protect your skin by wearing gloves. Look at the second image. In the second image, the TLC plate runs samples which are too much concentrated. A spot will be streaked and run together. If this happens, you will have to start over with a more diluted samples to spot and run on a TLC plate. Now look at the third image. Here what overloaded plates look like compared to well spotted plates. The plate on the left has a large yellow smear. This smear contains the same 2 components which are nicely resolved on the plate next to it. Now the calculation or evaluation part of the thin layer chromatography. For that we are using RF value determination. RF is a retention factor. It is the distance travelled by compound divided by the distance travelled by the solvent. So as an example, if compound is travel 2.1 centimeter and the solvent front travels 2.8 centimeter, then RF value will be 0.75. The RF of a compound is a constant from one experiment to the next only if chromatographic conditions are like solvent system is constant, adsorbent is constant, thickness of the adsorbent should be constant, amount of material also should be constant and temperature should be constant. In this kind of techniques, there are always some troubleshooting have observed. So TLC is quite an easy procedure, but what about the first time you have run the TLC and you see spot everywhere and blurred streaked spot? As with any techniques, with practice you get better. So as an example of common problems encountered in TLC are mentioned here. First is the compound runs as a streak rather than a spot. Then in this kind of condition, sample was overloaded. Run the TLC again after diluting your sample, or your sample might just contain many components creating many spots which run together and appear as a streak. Perhaps the experiment did not got as well as expected. Second is the sample run as a smear or upward crescent. In this condition, compounds which possesses strongly acidic or basic groups sometimes shown up on the TLC plate with this behavior. Add a few drops of ammonium hydroxide or acetic acid to the eluting solvent to obtain clear plates. Third is the plate solvent front runs crookedly. Then in this condition, either the adsorbent has flaked off the side of the plate or the side of the plate are touching the side of the container or the paper used to saturate the container. As the plate develops, crooked plates make it harder to measure RF values accurately. Next is many random spots are seen on the plates. Make sure that you do not accidentally drop any organic compound on the plate. If get a TLC plate and leave it lying on your workbench as you do not experiment, you might drop or splash an organic compound on the plate. Next is you see a blur or a blue spot on the plate as it develops. So perhaps you used an ink pen instead of a pencil. So you can see this kind of problems. Last is no spot are seen on the plate. This is the condition when you might not have spotted enough compound, perhaps because the solution of the compound is too diluted. Try to concentrate the solution or spot it several times on the place, allowing the solvent to dry between applications. Some compounds do not show up under UV light. Try another method, visualizing the spots such as staining or exposing the iodine paper. Or perhaps you do not have any compounds because your experiment did not go as well as planned. If the plate, the solvent will dissolve the compounds into the solvent reservoir instead of allowing them to move up the plate by capillary action. So you will not see spot after the plate is developed. These photos shows how the yellow compound is running into the solvent when lifted from the developing jar. So these are some problems which occur during the TLC process. Thank you for watching this session.